The third part of notes for Unit 5 is about enzymes and activation energy. So we talked about ATP and how that forms, and what you need to realize is that most chemical reactions don't occur just on their own. They require a little bit of energy to get started. We call this activation energy. It's kind of like when you're going to ride your bike or your skateboard down that hill, you can't just let go and start rolling. You have to kind of push off a little bit. And so that's kind of what activation energy is. There is a barrier that's required. You have to reach a certain amount of energy before the reaction will occur, and we call that the activation energy barrier. So how do cells overcome that? They overcome that by something called enzymes. We've mentioned enzymes before, of course. Here's a definition for you. Enzymes are a biological catalyst that speed up chemical reactions by lowering the activation energy of the reaction. So rather than having that high barrier that we saw before of energy, the enzyme makes that barrier a little bit lower. Most enzymes are made of proteins. There are a few other kinds of enzymes that are not proteins, but most enzymes in living things are proteins of one kind or another. Another characteristic of catalysts is they are not used up in the reaction. So they can be used over and over again without wearing out. Another characteristic of enzymes is they are very specific for a reaction. They're called substrate specific. The name of an enzyme generally ends in ASE, like a sugar ends in OSE. So instead of glucose, the sugar, we would have glucase if we had an enzyme that broke that down. As far as I know, there isn't such an enzyme, but that's what it would be called. An enzyme generally works best at a certain temperature or a certain pH. The temperature in animals is generally about the body temperature of the animal. In plants, it's about the normal temperature of the plants, maybe a bigger range in plants because they live in a different range of temperatures than your body generally does. And they work at a certain pH. So some work better at a more acid pH and some work better at a more basic pH. Some enzymes require these non-protein helpers called cofactors or coenzymes, and we'll mention some of those along the way as we're talking about some of these reactions. So how do enzymes work? Well, here's what happens. Each enzyme has a specific shape, and the shape is going to help make the chemical reaction happen. There's an opening or a place on the enzyme molecule where the substrate or the reactant can fit, and that's called the active site. The substrate is the molecule that the enzyme is going to work on. In this case, it's sucrose, which is table sugar, and it's going to fit together right into that active site in exactly the way. When it fits into that spot, it's going to make the enzyme change shape a little bit to make it easy to break that bond and add the hydrogen, and so the hydrolysis reaction is going to occur here. And the products are going to be released, glucose and fructose, which make up sucrose, and then we end back with our enzyme that looks exactly like it did to begin with. Okay, so the substrate fits into the active site, the enzyme is going to catalyze or make easier the reaction, and then the products are going to be released and the enzyme is available to be used again. Now, cells can control enzymes in several different ways, and we're going to do a couple of activities in class illustrating how this works. But one way that enzymes are controlled is by increasing the concentration of the enzyme. The more enzyme there is, the more reactions can occur. Another thing that can control the action of the enzyme is the substrate concentration. If you have too much substrate, then the reactions are going to slow down somewhat if you don't have enough enzyme. Another way that they are controlled is something called competitive inhibition. In competitive inhibition, you've got an inhibitor that fits into part of the active site but blocks the substrate from attaching there. Since it kind of looks similar to the substrate, the enzyme doesn't really care what it is as long as it fits in place. And if it prevents the substrate from attaching, then the substrate cannot be broken down or built up. And so it basically competes for the active site with the substrate, which makes it a competitive inhibitor. Now let's look at a couple of graphs and see if we can figure out which one is endergonic and which one is exergonic. Remember, an exergonic reaction is one that releases energy, and endergonic is one that requires or absorbs energy. So this would be over here on the left an energy absorbing reaction because the reactants start off at a lower potential energy than the products do. So this would be an endergonic reaction because it absorbs energy. This one, the reactants that are at a higher potential energy than the products, and so this is an energy releasing reaction. And of course, to reduce that activation energy barrier, if we have an enzyme, that will make that exergonic reaction happen more easily, more quickly, and that's going to be catalyzed. When it's catalyzed by the enzyme, it's going to make it work more quickly. We will talk a lot more about enzymes and do some activities about enzymes in class, but this concludes the notes for this unit.